Okay, can somebody please confirm that you can hear me?
Yeah, I can hear you.
Thank you. All right, before we begin, are there any questions? Anything? So we will begin as soon as I open my screen, share my screen, optimize for video. There we go. All right. Let's see if I can find where we were. Oop, there. Yep. So today is May 8th, and we're supposed to be finishing chapter 5 today and starting chapter 6. There is a lab today, lab 7. Any questions about what we're doing? All right. So we had talked about the generation of ATP and how it can happen by substrate level phosphorylation, where a phosphate group is removed from a molecule and added to ADP to make ATP. It can also be made in respiration, where ATP will be made. This doesn't show you how ATP is made, but it can happen in respiration, and ATP is made in various steps so that the energy from the electrons coming from glucose are some of that energy is given off to NADH, which some of that energy is given off when the electron FMN, and then the electrons pass to Q, etc., down the chain. At each step, the electron gives off some energy, and in cells, this is how our cells can survive in the making of ATP, because if we were to burn glucose to make ATP directly in one step, there would be so much energy given off that it would fry our cells. So the energy from glucose comes off in steps, and that way it can be done at 37 degrees, the temperature of your cells. So the second way of making ATP is respiration. And then the third way of making ATP is photophosphorylation. Photophosphorylation only occurs in photosynthetic cells. Light, light releases electrons from chlorophyll, and the light energy can be converted to chemical energy via an electron transport chain. In the process, ATP and NADPH are generated. However, they are often used to make sugar, so that frequently the end product of photosynthesis is sugar. And this picture is showing you some photosynthetic organisms, why the buoys are so muddy. Any question about any of that? All right, that's a little bit of a joke. There's photosynthetic organisms in the background, but they're not photosynthetic. I also take an exception to here, and I just realized this. This is how photosynthetic processes happen in most cells, but uh, light can release electrons from another photosynthetic pigment. It doesn't have to be chlorophyll. Like red algae and brown algae have red pigments or brown pigments, and they can engage in photophosphorylation and photosynthesis, but it's not using chlorophyll. Chlorophyll is the most common pigment to release the electrons from light, or light releases the electrons from the photosynthetic pigment chlorophyll. All right, let's talk a little bit about the metabolic pathway of energy production. Energy is released from molecules in controlled ways and stored by a series of reactions, meaning the energy in cells is not released in a single energy burst. The electrons from donor molecules are passed from one compound to another through a series of redox reactions. And I've already showed you those redox reactions right here. And ultimately, it's not showing you the electrons generally generate from glucose. They're passed to NADH. Then they're passed through the chain of the proteins in the electron transport chain, and we'll talk a little bit about that later. So the electrons from the donor molecule, in this case glucose, are passed from one compound to another. Through a series of redox reactions, the energy is not released all at once. Carbohydrate catabolism is a frequent way that organisms use glucose for their energy needs. So carbohydrate catabolism is the primary source for cellular energy. The most common energy source is glucose, and as you know, our cells can burn glucose to create ATP to give us energy. But other catabolism can be done. Our cells can also use lipids and proteins and other carbohydrates, obviously. But glucose is the preferred way. And generally, for most of us couch potatoes, whatever we eat, that molecule will be converted into glucose. And then glucose will be shunted down the pathway to make ATP. So if you ate a high lipid diet or a high protein diet, you can burn those to make ATP. To produce energy from glucose, microorganisms use two general processes. They use respiration and they use fermentation. Of respiration, there are two. Aerobic respiration. This is the form of respiration that we can perform, and it's very common in many organisms like E. coli, Staphylococcus epidermidis. What would be one that only uses aerobic respiration? Pseudomonas aeruginosa, maybe? Another form of respiration is anaerobic respiration. This does not use oxygen. And obviously an anaerobe would use anaerobic respiration. Aerobes use aerobic respiration. And some organisms use fermentation. Your muscle cells can use fermentation. And then Streptococcus, what is it? No, Lactobacillus, as well as Streptococcus, something that makes yogurt. Those organisms only use fermentation. The yeast making bread or beer uses fermentation. Any question about any of that? If not, let's watch a little video on aerobic respiration. Well, it's taking a while to download.
All cells need energy.
I'm going to turn the volume up.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with the molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into 2 molecules of pyruvate, energizes 2 electron carrier molecules, and produces 2 molecules of ATP. The next 2 stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create 2 more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
All right, any questions about that video? We'll talk about aerobic respiration in great detail in this lesson. So here we're comparing aerobic respiration to fermentation. There are 3 principal stages of aerobic respiration. The first, glycolysis. The second, the Krebs cycle. And we could call it the preparatory step and the Krebs cycle. And then the last step, electron transport chain. Fermentation only has glycolysis. There is no Krebs cycle. There is no electron transport chain. Essentially, in aerobic respiration, the electrons flow from glucose, an energy-rich molecule, to carbon dioxide and water, which are energy-poor molecules. In fermentation, the energy flows from glucose to pyruvate, and in the process of aerobic respiration, we get a high ATP yield. In the process of fermentation, the pyruvate is converted to the end product of fermentation, and there are different end products depending on which fermentation pathway we're using, but the ATP production is much less, much lower in fermentation than in aerobic respiration. Any questions about this overview? All right, this is showing you an overview of aerobic respiration in a eukaryote cell. Aerobic respiration has 3 parts: glycolysis, the Krebs cycle, and the preparatory step, and then the electron transport chain, which is right there, electron transport chain. Initially, in a eukaryote cell, the glucose is converted into pyruvate in the cytoplasm. So if we look at the pink boundary, that's the boundary of the cell. Glycolysis occurring in the cytoplasm. Pyruvate is made by glycolysis. Some ATP is made. Some NADH is made. We'll talk about the NADH in a bit. And then the pyruvate moves inside the mitochondria. Moves inside the inner membrane of the mitochondria. The pyruvate is converted and then joins the Krebs cycle. How that happens is the preparatory step. We'll talk about that in a little bit. And the Krebs cycle is also called the citric acid cycle or the TCA cycle, or one other name too, which escapes me at the moment. In the process of the Krebs cycle, it's named the Krebs cycle because Dr. Krebs discovered it. It's called the citric acid cycle because citric acid is one of the first molecules in the Krebs cycle. In the Krebs cycle, some ATP is made, some more NADH and FADH2 are made, and the carbon in glucose, or converted to pyruvate, gets all burned into CO2 in the preparatory step and the Krebs cycle, shown here and here. Some ATP is made. The NADH and the FADH2 carry the electrons that ultimately came from glucose to the electron transport chain, or the third process of aerobic respiration. So that's what these NADH and FADH2 are doing. They're carrying electrons to the electron transport chain. Now, this Krebs cycle, or citric acid cycle, happens inside the matrix of the mitochondria of a eukaryote cell. The electron transport chain occurs across the inner membrane of the mitochondria. The electron transport chain has a few other names. One of them is oxidative phosphorylation. I will always use electron transport chain, which is the term your textbook tends to use as well. In the process of the electron transport chain, we make a lot of ATP. In the electron transport chain, the electrons being carried by NADH and FADH2 are given to the electron transport chain, and the electrons move from one molecule in the chain to another to generate ATP. Any question about any of that? Now remember, this is an overview of aerobic respiration in a eukaryote cell. Prokaryote cells do not have a mitochondria, and so in a prokaryote, glycolysis occurs in the cytoplasm. The Krebs cycle also occurs in the cytoplasm. The electron transport chain occurs across the cell membrane. So it's not the inner mitochondrial membrane in a prokaryote cell, it's the cell membrane. So when we're talking about aerobic respiration, the first step is glycolysis. Glycolysis does have another name, which I do not use. I think your textbook mentions it once, the Embden-Meyerhof pathway, because these scientists discovered glycolysis, and so that's named that sometimes. I'm not German, so I could be butchering their names. In glycolysis, glucose becomes oxidized and converted to pyruvic acid. Glycolysis can occur in most living cells, and glycolysis can occur in the presence or in the absence of oxygen. Glycolysis does not use oxygen. It does not need oxygen, but it can occur in the presence of oxygen. Glycolysis does produce some ATP and some NADH. Any question about any of that? All right. Here we're looking at the different steps of glycolysis. Let me blow this up. There, now you can see it better. Glycolysis has some steps, 9 or 10 steps, where I can't get that out of here. Well, we'll start there. Where glycolysis, oh, that's good. Starts with glucose. Why there's 6 circles here starting, oh, there's glucose there. And you actually have to put in 2 ATP molecules, right here and right there, to get the process running. And then you make glyceride 3 phosphate and DHAP, or dihydroxyl acetone phosphate. But the dihydroxyl acetone phosphate can be converted into glyceride 3 phosphate. So that's why this pathway, you can either go here or go there, doesn't matter which. But here it's going here and then there, and then you go down. The this molecule here, we're just going to call it that because you don't need to know the molecule, the name of it here, is converted into this molecule, making an NADH, two of them actually, and then making two ATP molecules in the next step. And then we go on down further, making water, and then making ATP, two molecules again here. And then we end up at pyruvic acid. Pyruvic acid is a three carbon molecule. Glucose is a six carbon molecule. So you make two pyruvic acids during glycolysis when you're using glucose. Any question about any of that? Now, when I was an undergraduate and I was learning glycolysis, I had to know the names of every molecule on this chart, and I had to know the enzymes too, which aren't even in here. It's just 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 10 steps, counting this one here. And I think it's fair that I had to know these. I think it's fair that you guys need to know these. What do you say to that? Come on. Oh, I know, in the show. Nobody objects? Okay, you have to know the names of the intermediates there.
Wait, I object.
Ah, okay. You only need to know the summary of glycolysis. You start with glucose, and you probably should know that you add in 2 ATP, but you get out 4 ATP, and that's a net gain of 2 ATP. You must know that you get a net gain of 2 ATP from glycolysis. You start with glucose, you end up with 2 pyruvates and 2 NADHs. We'll talk about the 2 NADHs in a little bit, although I've already talked about them. And then a word of caution, your cells, as well as us in microbiology, will convert pyruvic acid and talk about it like pyruvate because your cell will use these two the same. The difference is pyruvic acid has a hydrogen on it. Pyruvate has the hydrogen that's come off of it, so it's lacking the hydrogen ion. And like I said, your cell uses these the same. A chemist may object, but we will use these terms to mean the same thing. So glucose can be made into pyruvate or it can be made into pyruvic acid. Any question about any of that? And your cell will use them the same. All right, after glycolysis, we then go on to the Krebs cycle. Glycolysis happens, as I stated, in a eukaryote cell in the cytoplasm, as well as in a prokaryote cell in the cytoplasm. In respiration, you should know that there are two forms of respiration. Respiration is the oxidation of molecules such as glucose, releasing electrons, mainly for the electron transport chain. There are two forms of respiration, aerobic respiration, which aerobes use, and anaerobic respiration, which anaerobic organisms use. In aerobic respiration, the final electron acceptor is oxygen, meaning the electron from glucose. In aerobic respiration, they flow to oxygen, and that's the last molecule to accept the electrons. Why it's called the final electron acceptor. In anaerobic respiration, the final electron acceptor is a molecule other than oxygen. It depends on which form of anaerobic respiration we're talking about. It could be nitrate, it could be nitrite, it could be sulfate, It could be some form of iron. And there are different anaerobic respirations, slightly different pathways. And the big thing is they have a different final electron acceptor. That's why it's called nitrate anaerobic respiration, because the electron flows to nitrate. Nitrite, the electron flows to nitrate. Sulfate, the electrons flow to sulfate. But the important part to remember is anaerobic respiration. The final electron acceptor is a molecule other than oxygen. Any question about that? In respiration, whichever form, ATP is generated. All right. So we have pyruvic acid or pyruvate coming out of glycolysis. That molecule, pyruvic acid, has to be changed so that it can directly enter the Krebs cycle, the next step, which the preparatory step is a part of the Krebs cycle, depending on how you define the Krebs cycle. But it's one step, changing the pyruvic acid to acetyl coenzyme A. In the process, NADH is made, and one of the carbon comes off pyruvic acid to form CO2. So that'll be the first carbon of glucose that forms CO2. And then the end result is acetyl coenzyme A. After the coenzyme A attaches to the remainder of pyruvic acid, and then the acetyl coenzyme A can directly enter the Krebs cycle. Two molecules of acetyl coenzyme A form from one molecule of glucose, because in glycolysis, one molecule of glucose makes one molecule of pyruvic acid, two molecules of pyruvic acid, and that's why the preparatory step will run twice for each molecule of glucose. Any question about any of that? All right. So the next step is the Krebs cycle. The Krebs cycle is also called the tricarboxylic acid cycle because there's a lot of tricarboxylic acids. This just means 3 carbon acids in the Krebs cycle. It's also called the citric acid cycle because citric acid is the first tricarboxylic acid in the Krebs cycle. And it's frequently shortened to TCA, which is a short name for tricarboxylic acid, or T for the citric acid cycle. We will call it the Krebs cycle, and your textbook tends to call it the Krebs cycle as well. So this is showing you the preparatory step, the one-step reaction before the Krebs cycle. Let me blow the slide up. So we had pyruvic acid. It's changed into acetyl coenzyme A by the preparatory step. We get one molecule of carbon dioxide being generated, one molecule of NADH being generated in the preparatory step. The acetyl coenzyme A then binds to the oxaloacetic acid of the Krebs cycle to make citric acid. You don't need to know that, by the way, which is converted into this molecule, converted into that molecule, making another molecule of NADH and another molecule of CO2, which is then converted into that molecule, making another molecule of NADH and another molecule of CO2, which is then converted into that molecule, which makes ATP in this step. And then this molecule is converted into that molecule, making a molecule of FADH2. This molecule is converted into that molecule. This molecule is converted into that molecule. Okay, it's warning me my internet connection is unstable. So if you don't hear something, please let me know and I'll repeat it. Did you hear all of the last? All right, not hearing anything. Anyways, when we take this molecule to make that molecule, we generate a molecule of NADH. And then this molecule is oxaloacetic acid, the molecule we started with. Why it is called a cycle? Because this cycle keeps repeating in the cell as long as we have acetyl coenzyme A coming in. The Krebs cycle will perform in your cells. And that happens for as long as you are alive. Any question about any of that? All right, so that's why it's called a cycle. In the process of the Krebs cycle and the preparatory step, we make one ATP molecule here, 4 NADH molecules, 1, 2, 3, 4, 1 FADH2 molecule, and then 3 CO2 molecules, 1, 2, 3. And that's for each pyruvic acid molecule, but realize when we burn glucose, you're going to run the preparatory step in the Krebs cycle 2 times because glycolysis makes 2 pyruvic acids from glucose. And so you have to be careful of how I word a question, and that is if I word it like how many ATP are made for pyruvic acid, you're going to say 1. But if I word it how many ATP are made from glucose in the preparatory step in the Krebs cycle, you have to double these numbers here, 2 ATP, 8 NADH, 2 FADH2, and then 6 CO2. Any question about any of that? All right, the NADH and the FADH2 carry the electrons that ultimately came from glucose to the electron transport chain, the third part of aerobic respiration. So this is once again showing you the overview of aerobic respiration and fermentation. So far, glucose being made into glycolysis, we've only generated a net of 2 ATP. And in the preparatory step in the Krebs cycle, per molecule of glucose, we also made 2 ATP. So that's a total of only 4 ATP so far. So that means the rest of the ATP is made in the electron transport chain, the third step of aerobic respiration. And in this step, ATP production goes into high gear, meaning we make a lot more ATP in the electron transport chain than we do everywhere else in aerobic respiration. Any question about any of that? Let me end the show is what I'm trying to do. All right. So the electron transport chain is the third step of aerobic respiration. It's sometimes called ETC for electron transport chain. It can also be called electron transport phosphorylation because ATP is made from ADP. So the ADP added on a phosphate group, and that's phosphorylation. But we will use the term electron transport chain. It does have other names like chemiosmosis, and what was the other one? No, I've forgotten now. I don't use the other terms because they're too many and too confusing. I will always use electron transport chain. Any question about that? All right, electron transport chain is a series of carrier molecules that are in turn reduced and oxidized as electrons that ultimately came from glucose are passed down the chain. Energy is released as electrons are transferred from high energy to lower energy compounds, meaning in the chain here, on the left side, we have the higher energy, and then on the right side, we have the lower energy. Energy is released and used by chemiosmosis to produce ATP. Any question about any of that? And chemiosmosis is just a part of the electron transport chain. There we go. So let's watch a little video on aerobic respiration and the electron transport chain.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
All right, any questions about that video? All right, let's talk about the electron transport chain in great detail now. The electron transport chain is called that because it is a chain of proteins in a membrane. And the electrons transfer from one protein to the next protein in the membrane. So why it's called the electron transport is because it's electron transported from one protein to another. And then it's called the chain because there's a chain of these proteins in a membrane. In eukaryotes, the electron transport chain is in the inner mitochondrial membrane. In prokaryotes, this chain of proteins in a membrane is the cell membrane. Any question about any of that? All right. In the process, hydrogen ions are pumped from this side of the membrane to that side of the membrane. And in a eukaryote cell, these hydrogen ions will be pumped in between the inner membrane and the outer membrane, so that in eukaryotes, we kind of protect the cell from these hydrogen ions by limiting them inside the inner to the outer membrane. In a prokaryote cell, these hydrogen ions are passed from inside the cell to outside the cell. And what is outside the cell membrane of a prokaryote cell? I'm trying to illustrate a point, and that is, where are these hydrogen ions being pumped in a prokaryote cell? Anyone have a guess? What's outside the cell membrane of a prokaryote cell? Nobody has a guess? Nobody knows? You'd fail a quiz question if I were to ask that on a quiz. The cell wall is outside the cell membrane of a prokaryote cell. So these hydrogen ions in a prokaryote are being passed inside the cell wall. Any question about any of that? In the electron transport chain, ATP molecules are produced at high levels in aerobic respiration. In eukaryotes, you make in the electron transport chain either 32 or 34 ATP molecules per molecule of glucose. Prokaryotes are a little more efficient. They always generate 34 molecules of ATP per molecule of glucose. In aerobic respiration, the final electron acceptor is a molecule of oxygen. The oxygen combines with the electrons and some hydrogen ions to form water. So here we're looking at the overview of aerobic respiration and fermentation. And there we're looking at the overview of electron transport, where the molecules which come from glucose are carried by NADH or FADH2, brought to these molecules. And then these are the molecules here, by the way. The electrons carried from one molecule to another, giving up some of their energy. And in aerobic respiration, the electrons ultimately flow to oxygen. Let me blow that up. The electrons ultimately flow to oxygen, forming water. And then we'll talk about this in a little bit. So there's that one there. This is how the water gets used up, by the way, by your cells, or gets made by your cells from aerobic respiration. And that is the electrons being carried by NADH or FADH2, which isn't shown here, but I believe it comes in here. Let me see if I can find that. It's going to Q. There's Q there. No, I guess they go right here. Anyways, NADH gives the electron to the first chain of proteins in the membrane. The electrons flow through this protein, and that forms NADH and an H plus ion, by the way, when the electrons are passed from NADH to the first protein in the chain. And then the electrons move to the next protein in the chain of proteins, to the next, to the next, to the next, to the next. We'll get to it. Excuse me, not to the next, and then to oxygen. And when the electrons flow to oxygen, Oxygen combines with hydrogen ions to form water, and that is where the electrons from glucose end up at. They flow to oxygen, so oxygen is the final electron acceptor in aerobic respiration. And that is why we must use oxygen, why we must breathe oxygen. All of that oxygen is converted into water in aerobic respiration. You should make note of that. None of that oxygen is converted into CO2. All of it is converted into water in aerobic respiration, shown here. Along the way, as the electrons flow through this protein and this protein and that protein, they give this protein and that protein and that protein the energy the protein needs to pump one hydrogen ion from this side of the membrane to that side of the membrane. And that's going to happen in aerobic respiration 3 times with that protein, and this protein. For NADH, I should say it happens 3 times. For FADH2, it only happens twice because FADH2 does not give its electron to the first protein in the membrane. Along the way, hydrogen ion is pumped from this side of the membrane to that side of the membrane. That creates a twofold gradient. There's more hydrogen on this side of the membrane than on that side of the membrane. There's also more positive charge because each of these hydrogen has a positive charge on this side of the membrane than on that side of the membrane. So these hydrogen ions are, by the laws of thermodynamics, going to want to move from this side of the membrane to that side of the membrane. But hydrogen cannot get through the cell membrane, cannot flow through the lipid bilayer, except at this channel protein, which is an ATP synthase enzyme. And when hydrogen flows from this side of the membrane to that side of the membrane, for each hydrogen flowing across this channel protein can make 1 molecule of ATP for each hydrogen ion coming across the channel protein. So it's phosphorylating ADP by combining phosphate to it to make ATP. And that is how our cells make, or at least most of our cells make most of their ATP. There is an exception. Your muscle cells can make ATP by fermentation, and we'll talk about fermentation later in this lesson. Any question about any of that? All right. So as I mentioned, in eukaryotes, the hydrogen ions are being pumped from the inner mitochondrial membrane inside the space between the outer membrane and the inner membrane. In the prokaryote cell, the hydrogen ions are pumped across the plasma membrane, and if the cell has a cell wall, they're being pumped into the cell wall. The hydrogen ions then flow across the membrane through this ATP synthase protein to make ATP. The electrons end up at oxygen in aerobic respiration to form water. Any question about any of that? All right, let me mention that the proton gradient has 2 forms of potential energy. There's more hydrogen on this side than on this side. There's more positive charges on this side of the membrane than that side of the membrane. So these hydrogen ions are going to want to move to this side of the membrane. That's potential energy, but potential energy is used to make ATP. And how that's happened is the hydrogen just flow through this channel protein, which for each hydrogen ion flowing through it can make ATP by phosphorylating phosphate and ADP. to make ATP. And that's specifically called chemiosmosis. You don't need to know that. There's another name for the electron transport chain, oxidative phosphorylation. It's oxidative because, well, the molecules are oxidized, and phosphorylation because ADP becomes phosphorylated. But we will call it the electron transport chain, not oxidative phosphorylation and not chemiosmosis. I'll just call it the electron transport chain. So in aerobic respiration, we get ATP made. So let's account for all the ATP that's being made. For complete aerobic respiration of glucose, eukaryote cells generate 36 to 38 ATP molecules. Prokaryote cells always generate 38 ATP molecules. They're a little more efficient. Now, coming up to the eukaryotes, most of the cells in your body can only generate 36 molecules of ATP per molecule of glucose. But your heart, your liver, and your kidneys are more efficient, and they can generate 38 ATP molecules per molecule of glucose. Why do you think your heart, your liver, and your kidneys are more efficient than, let's say, your skin cells, or your blood cells, or right now your brain cells? Why is your heart more efficient than your brain right now? Come on, somebody's got to have a guess. I'm saying your brain because you're obviously not thinking, so you're not using ATP, meaning your brain is not always working. And that's why you can't come up with the answer here. But your heart, is it always working? That's a yes or no question, people.
Yes.
Yes.
Always working? Yeah, as long as you're alive, your heart is always working. So the heart is more efficient because it's always working. Most of the cells in your body are not always working, but your liver cells always are, and your kidneys always are too. The kidneys have to recycle the blood, purify it. The liver is always removing toxins from the blood. The toxins are delivered to the liver and then the liver. So the point is the liver, the kidneys, and the heart are always working for as long as you're alive. And then your skin cells, most of the time, they're not working. Okay? And that's why the heart, the liver, and the kidneys are more efficient than all the other cells in your body. All right. So in the summary of aerobic respiration, we start with glucose, C6H12O6. We add in 6 oxygen molecules, 38 ADP molecules, 38 phosphate molecules, and then out of this, we get 6 carbon dioxide, 6 water molecules, and 38 ATP. You should know the summary equation for making ATP and electron in the aerobic respiration. You start with glucose, and you get out of it 6 CO2. None of these CO2 come from oxygen. All these CO2s come from glucose. And then the same with the water. All of the water comes from the oxygen. And the ATP comes from ADP. Any question about any of that? All right, to remind you, glycolysis makes 2 NADH and 2 ATP. The Krebs cycle in the preparatory step, make 2 more NADH and 2 more ATP. And then the electron transport chain generates 34 to 32 ATP molecules for a total of 36 to 38 ATP for aerobic respiration. And then the NADH, can't read that, 6 and the 2 FADH2 are converted into the electron transport chain to make water. And ATP, obviously. All right, any question about that? You should know the numbers. You need to know the numbers for ATP, glucose, pyruvic acid, acetyl coenzyme A. I don't think I ever asked you about the NADH, but you probably should know NADH as well. And then you should know that when you burn glucose, you will get 6 carbon dioxide molecules made because glucose is a 6-carbon molecule. Carbon dioxide is a 1-carbon molecule, and all of the carbons in glucose are oxidized into CO2. All right, any questions about aerobic respiration? If not, let's talk a little bit about anaerobic respiration. The main difference between anaerobic respiration and aerobic respiration is the final electron acceptor is a molecule other than oxygen for anaerobic respiration. It can be a different molecule depending on which form of anaerobic respiration we're talking about. Like I said, it can be nitrate, it can be nitrite, that'd be NO2 minus. It can be sulfate. In anaerobic respiration, glycolysis and Krebs cycle still occur. The proteins in the chain of the electron transport chain is a little shorter. So let me show you that again. Right here. It's a little shorter chain, so instead of being 1, 2, 3, 4, 5, instead of, well, 6, we count that one. Instead of being 6, it'll be shorter. Depending on which form of anaerobic respiration it is, it can be a lot shorter or it can just be 1 protein shorter. Because the electron transport chain is shorter, we do generate less ATP than aerobic respiration. The ATP will be something between over 2 to 36 molecules of ATP. Because anaerobes generate less ATP than aerobic respiration, anaerobes tend to grow slower than aerobes. Any question about any of that? Remember, with a prokaryote in aerobic respiration, you get up to 38 ATP. So for prokaryotes, anaerobic respiration always generates less ATP than aerobic respiration. And that makes anaerobes tending to grow slower. Any questions about anaerobic respiration? All right, let's move on to fermentation. What we've compared aerobic respiration to is fermentation. Fermentation always involves glycolysis as the first step of the fermentation process, and then 1 or 2 fermentation steps after glycolysis. In fermentation, you only generate 2 ATP during glycolysis, a net gain of 2 ATP per molecule of glucose. The fermentation steps will convert pyruvic acid to the end product of fermentation. And there are different end products depending on which fermentation reaction we're talking about. So ATP production and fermentation is not very much. We only generate a net gain of 2 ATP per molecule of glucose. And all of those ATP are generated in glycolysis. We do generate some NADH and H+ from the oxidation of glucose, but that NADH is used up not to generate ATP, but to perform the fermentation step of fermentation. So the NADH will combine with pyruvic acid to make the end product of fermentation. Now, the end product could be alcohol, or it could be, meaning ethanol, or it could be lactic acid. The end product will depend on which fermentation reaction we're talking about, and there's about 6 of them. Fermentation does not use the Krebs cycle or the electron transport chain, but it always uses glycolysis. And then the electron flows to an organic molecule as the final electron acceptor. In this case, pyruvic acid is the final electron acceptor. You don't need to know that, but you just need to know that it's a molecule of the fermentation process. There are 2 fermentation reactions we're going to talk about: lactic acid fermentation, where glucose will end up as lactic acid after fermentation. You generate 2 ATP and glycolysis. You generate 2 NADH in glycolysis, but the NADH is used to convert the pyruvic acid to lactic acid. Lactic acid fermentation happens a lot. We use it to help prevent food spoilage. We use it to make sauerkraut, yogurt, a whole bunch of cheeses like cheddar cheese, a whole bunch of other foods. I'm drawing a blank here on, well, sausage would be one. Oh, I guess that's, yeah, it's not there. And then chocolate, another I happen to know. Your muscle cells can perform lactic acid fermentation, converting glucose into lactic acid. We'll also talk about alcoholic fermentation. It's a 2-step process, converting pyruvic acid into this molecule here, which is a 2-carbon molecule. Pyruvic acid is a 3-carbon molecule, and that gives off CO2. If you were to cap the bottle of beer, that CO2 gives the beer its fizz. Wine also uses alcoholic fermentation, but it's made in an open barrel or casket, and so the CO2 vents to the air. If you were to make the wine in a bottle and cap the bottle, we would call it champagne, from the bubbles being produced by the CO2 of the alcoholic fermentation. Anyways, this molecule is then converted by NADH into ethanol to make alcoholic fermentation. Now, why do your cells use up NADH to make lactic acid when the NADH could be stored in the muscle cell and then used in the electron transport chain, and you get a lot more ATP out of the electron transport chain than you would get out of fermentation, which for this fermentation step, you don't get any ATP out of it. You get it right here in glycolysis. So why does your muscle cell use up that NADH to make lactic acid rather than store it and then use that NADH for aerobic respiration? Anyone have a guess? Give you an extra credit point if you get it right. It's actually shown in the slide here.
Can you repeat that question?
Why do your muscle cells use up the NADH to make lactic acid, which makes no ATP, instead of store the NADH to use in an electron transport chain to make ATP?
Would it still be for energy, even though it's not making ATP?
Kind of. The NADH in making lactic acid makes NAD plus, recycling the NAD plus so that glycolysis can continue to happen. If you run out of NAD plus, glycolysis will end because you need NADH plus to get glycolysis to occur. So the cell, when it's low on oxygen, the muscle cell, and the muscle cells only, can perform lactic acid fermentation, taking this NADH being made by glycolysis to make lactic acid, and that will allow glycolysis to continue in being done in the cell, which will make some ATP. And your muscle cell will want ATP because once you have no ATP, the muscle can't work. And we've probably all seen this during the Olympics. There's usually one Olympic athlete who pushes themselves to the limit and they run out of ATP because they really run out of glucose in their cells. And when they have no ATP being made by fermentation, the muscle cannot work because it doesn't have any ATP, and the athlete just collapses. In the summertime, the coach will run out and spray them down with water. In the wintertime, the coach runs out and covers them with a blanket. And then the Olympic athlete has their heart and lungs working like mad to supply the muscle with blood. The blood will bring glucose and oxygen, and then the cells, the muscle cells, will get ATP from aerobic respiration. And then the Olympic athlete will stand up and walk across the finish line. Any question about any of that? Anyways, your muscle cells can perform lactic acid fermentation, but only when oxygen becomes limiting, meaning the muscles are working so hard, there's no more oxygen in the cell. There is some glucose. The glucose can be used up by fermentation, lactic acid fermentation, to make lactic acid. The lactic acid burns up in your muscle. It's an acid and it actually makes the muscle burn. You can feel it. And that way your muscles can continue working because some ATP is made, even though the muscle is short on oxygen. So it can't perform aerobic respiration. Any question about any of that? Now, once the athlete stops using the muscle, at least hard, meaning either using the muscle just a little bit or not at all, aerobic respiration will continue because the heart and the lungs are working like mad to bring oxygen to the muscle. And so oxygen will come back to the muscle, and then the muscle cell will start using aerobic respiration and stop using fermentation because aerobic respiration is much more efficient at generating ATP. Any question about any of that? And we'll talk about the lactic acid later, but it will be converted back to pyruvic acid and then go through aerobic respiration the normal way, meaning it'll perform the preparatory step in the Krebs cycle. All right, any questions about any of that? All right, this is showing you the different fermentation reactions. We've talked about lactic acid fermentation and alcoholic fermentation. Lactic acid fermentation can happen by Streptococcus and Lactobacillus, some bacteria used in yogurts to make yogurt, but a lot of different organisms can perform lactic acid fermentation, including your muscle cells. A yeast, Saccharomyces, can perform alcoholic fermentation, giving the bread leavening, meaning when we put yeast in our bread and get the raised bread. We can also use the yeast to make wine or beer, to make alcohol. Proponic acid, you don't need to know this, but that's another form of fermentation. And this one is the reaction to make Swiss cheese. You can note that Swiss cheese, proponic acid fermentation creates CO2. The CO2 being given off makes the holes in the Swiss cheese. Clostridium can make these products, sometimes used to make butanol, acetone, isopropyl alcohol. Salmonella, making ethanol, lactic acid, acetic acid. Enterobacter, making, I'm not seeing it here, but I think Enterobacter is one that makes vinegar. Anyway, some different fermentation reactions. Like I said, you only need to know the first two. Some of the different fermentation reactions, their industrial use or commercial use, like lactic acid fermentation, making rye bread. Most cheese, like Swiss cheese, not Swiss cheese, cheddar cheese and most other cheese, not Swiss cheese, yogurt, summer sausage, sauerkraut, chocolate, acetic acid making vinegar. It starts actually from using ethanol to make the vinegar. Acetobacter, I got that wrong. I was thinking it's Enterobacter. Proponic acid making Swiss cheese. And then some other products we make from fermentation. A good summary of aerobic respiration, anaerobic respiration, and fermentation is in this slide. So here we have aerobic respiration. The growth conditions have to be aerobic. Aerobic respiration requires the use of oxygen. So oxygen is the final electron acceptor. Anaerobic respiration does not use oxygen. Its growth conditions have to be anaerobic. The final electron acceptor is an inorganic substance other than oxygen, like nitrate, sulfate, carbonate, some molecule other than oxygen. Fermentation can happen either in aerobic or anaerobic respiration. Doesn't use oxygen, but it's not hampered by oxygen. The final electron acceptor is an organic molecule of the fermentation. And then you don't need to know this column. You should know the ATP molecules produced. Aerobic respiration is the most efficient. 38 ATP molecules for prokaryotes, 36 to 38 for eukaryote cells. For anaerobic respiration, it's always less than 38, so it's between over 2 to 36, so something like 4 to 36, depending on which form of anaerobic respiration we're referring to. Some of them are almost as efficient as aerobic respiration, but some of them are just a little more efficient than fermentation. Fermentation produces the least ATP. We only make 2 ATP molecules per molecule of glucose in fermentation. Any question about this slide? All right, so I've talked about glucose going through respiration, especially aerobic respiration, in the normal way, and then being burned to make ATP. Well, as you know, most other carbohydrates can be converted to glucose. So a carbohydrate can be converted to glucose and then go through aerobic respiration in the normal way. But lipids and proteins can also be catabolized. and they can be burned and used by aerobic respiration. However, there is a problem with lipid and protein metabolism, and that is proteins and lipids tend to be very large molecules, and these large molecules cannot cross the cell membrane. So how cells digest lipids and proteins, including your gut cells, is they will excrete, meaning produce lipases, that's something that digests lipids, and proteases, something that digests proteins, and then secrete those molecules extracellular and digest the lipid and the protein outside the cell. And bacteria will do this as well, creating the monomers of the lipids and the proteins, which are then small enough to come across the cell membrane. For proteins, the monomers will be amino acids. For lipids, it'll be fatty acids and glycerol. And then the monomers can be digested in aerobic respiration. Any question about any of that? So this is showing you protein digested. Let me see if I can blow this up. The proteins will be converted into amino acids. The amino acids can go in a late step in the Krebs cycle, depending on which amino acid it is. It can go and be converted into acetyl coenzyme A and then go in the Krebs cycle. It can also be converted into a late step of the Krebs cycle, depending on which amino acid it is. Lipids will always be converted into glycerol and fatty acids. The glycerol will go midway through the Krebs cycle. The fatty acids will be broken into 2 carbon chunks and then converted into acetyl coenzyme A and then enter the Krebs cycle. For lipids, your body cannot burn them, or let's say not your body, but a marathon runner cannot digest and burn lipids directly in aerobic respiration until the marathon runner is 45 minutes into their run. So the first 45 minutes, they will not burn lipids in aerobic respiration. But after 45 minutes in the run, they can burn lipids and use the burning of the lipids or using the lipids to make ATP. I don't know where a marathon runner would start digesting the protein. It may depend on what the amino acid is. But the point is, if you eat a whole high protein diet, you can burn the proteins and make ATP from digesting the proteins. So the complete metabolic oxidation of glucose in aerobic respiration is the most efficient way for making ATP, but energy from ATP can be made in other ways. Most cells will use this ATP for their energy needs. For example, the biosynthesis of a biological molecule. And like I said, most cells can also metabolize lipids, proteins, and nucleic acids, the 4 molecules of life, in aerobic respiration. But for the marathon runner, they cannot immediately digest lipids. It'd have to be 45 minutes into their run. All right, any questions about any of that? If there's no questions, I'm going to end it here and I'll see you in the lab at 6:30. Any questions? All right, I'll see you at 6:30.